Hello friends, so in today's lecture we are continue with our unit 6 in specially photosensory biology or photomorphogenesis. We earlier talked about the first photoreceptor, which is a phytochrome, which is responsible for perceiving red and far red light. Now we are going to talk about our second photoreceptor or photopigment, which is a cryptochromes, which is a blue light receptor. So this is something about my CSR note. If you are interested, you can contact me in this given number. Now cryptochrome, it is a chromoprotein or a flavoprotein. It means it is a merging of two things, which is a flavin group and a protein. It is a blue light photoreceptors. There are two types of blue light photoreceptor are present in plants, which is a cryptochrome and phototropins, which we going to talk about in our next video. Cryptochrome means hidden color. The chromophore for cryptochrome is a flavin and pterin. So these two groups are present as a chromophore. If we talked about the phytochromes, in that the phytochromobilin was the chromophore group. Now in cryptochrome, the flavin and pterin is a chromophore group. Three common type of flavins are there. Now the types of flavins, which is a riboflavin or its derivative, FMN and FAD. Flavin plus protein is equal to flavoprotein. So it is a merge of two things, flavin and a proteins. This means that cryptochrome is a flavoprotein. Now pterin group. It is a light absorbing pigment present in also insect, fish and birds, not only in plants. Photoreceptor associated with blue light responses. There are three types of photoreceptor, which is a cryptochrome, phototropins and zeaxanthin, which have less studied in case of zeaxanthin. Cryptochrome present in all plants, insect and also mammals. Now let's check the properties of cryptochrome. Structurally related to photolyase. Now remember photolyase, it is responsible for photoreactivation by perceiving blue light. It nullify the dimer TT dimer formation in DNA repair. So it is a formation, it is responsible for DNA repair, but it is a look like a photolyase, not a photolyse activity. So photolyse is a blue light activated enzyme that repair pyrimidine dimer in DNA, but cryptochrome, it is a look like or conserved sequence and structurally related to photolyse but don't have any activity of DNA repair. This you have to remember. Cryptochrome don't have DNA repair activity, but they are thought to have evolved from photolysis, play an important role in generation and maintenance of circadian rhythm. The circadian rhythm, it is a biological clock occurs inside our body, that where we are hungry, when we need sleep, this all is regulated by a specific time. It is called a circadian rhythm or a biological clock, in which for about plant, the cryptochrome, it is mainly responsible for maintaining and generation of different rhythms, which is a circadian rhythm. The main function, it is a help in anthocyanin production, which is a pigment. Stromal opening, this you have to remember, it is a mediate the stromal opening circadian clock. They are evolutionary old and highly conserved protein because they are thought to have evolved from a photolysis, which is evolutionary conserved. Phototropism is also done by this. Photomorphogenesis, obviously, light capture, inhibition of stem elongation, this you have to remember. So it is antagonist of gibberellic acid, because gibberellic acid is responsible for stem elongation, but the cryptochrome is responsible for inhibition of a stem elongation. Hence they both are antagonists to each other. Maintaining the circadian rhythm. Magnetoreception, this you have to remember, sensing of magnetic field in number of species, that how the where the magnetic fields or rays are coming from in plant. This is a unique concept called magnetoreception. If we go to the structure, then at N terminal the PHR domain is there, which means photolysis homology region, and in C terminal DAS domain is also present. Now it is in a dimer form. So with the PHR, the binding of MTHF, which is a methyl transfer lives is there, and FAD flavin group is also there. So pterin group and flavin group are bounded with the protein, and they are bound with the non-covalent bond. When they light capture, they bound with the I or covalently. Now, dimerization is essential for a, so they are separately before they are activated. So in inactivated form, they both are separated, but at the light comes when they are activated, they must have to bind and form a dimer, which is initiated by C terminal. The C-terminal domain transduces the light signal to interact with the E3 ubiquitin ligase COP1. Now remember the COP1 in phytochrome. We talked about the COP1. COP1 is nothing but E3 ubiquitin ligase, which is responsible for maintenance of the photomorphogenesis during the light reception, especially for red light. The PR to PFR form is I am talking about the phytochromes. If you didn't knowledge about that, so please watch my video about phytochromes so you have a clear idea but what I am talking right now. So PR to PFR form, as PR to PFR form is happen, so COP1 is activated and moves nucleus to cytoplasm. So in nucleus, HY5 is free and mediate the photomorphogenesis, but during the far red light or inhibition of photomorphogenesis, COP1 is inactivated and COP1 remains present into the nucleus and bind with the HY5, which is a transcription factor, and mediate its ubiquitin ligation and mediate the 26S proteasome degradation. Hence HY5 concentration is decreases and HY5 cannot able to bind with the DNA sequence and mediate the transcription, and hence photomorphogenesis is not occur. Same thing happens here. Now what happened? The cry1 bind to COP1 and SPA1 complex and prevents its degradation. Now I talked about the COP1. Now COP1 is activated during red light. So during other lights it is inactivated. Now the cryptochrome is a blue light receptor. It is mediated the same thing as phytochrome. So what happened? As the blue light perceives by cryptochrome, the cry1 is activated and cry1 go to nucleus and bind with the COP1, which is inactivated, and mediate the COP1 detachment from HY5. Now HY5 is free to mediate the photomorphogenesis. The simple concept is there. Now let's talk about the cryptochrome genes. Three types of cryptochrome genes is found in Arabidopsis thaliana, which is a model plant in case of any botanical studies. Cry1, Cry2 and Cry3. Cry1 and Cry2, it is a translated by nuclear gene, and Cry3 is translated by chloroplast or mitochondrial gene, in which Cry1 and Cry3 is light stable and Cry2 is light sensitive. Cry2 high when blue light stimulate and cotyledon expansion. So Cry2 will be high. So Cry1 is involved in the setting of the circadian rhythm. In so a special function of Cry1 is to maintain the circadian rhythm, and Cry1 and Cry2 play a role in induction of flowering in cryptochrome. So Cry1 and Cry2 both responsible for induction of flowering, and they both prevent the degradation of HY5 from COP1. Cryptochrome homologs have been found to regulate the circadian rhythm in Drosophila mouse and humans also. So if we go to the mutation cases, mutation in Cry2 delay the flowering under continuous white light. Cry1 was proposed to be a blue light photoreceptor mediating the inhibition of stem elongation, which is a normal function of Cry1. But when Cry1 is mutated, what happened? Normal growth in red and far red, but elongated hypocotyle in blue light. This you have to remember. Cry2 mutants is delay flowering because blue light promotes the flowering. So this was all about our cryptochrome. I hope you learnt well. Jai Hind, Jai Bharat.